Hello there and welcome back to the Sleep Science, where tonight we're slipping beneath your skin, not in a horror movie way, but in the literal microscopic sense. You're going to drift into the dim, pulsing world of your own cells. Imagine yourself shrinking down until the familiar scale of your bedroom dissolves, the mattress stretching like a continent, the air thickening into a slow soup. Suddenly, you're weightless, gliding through a body that's both yours and a vast alien cosmos. Here, countless tiny citizens are buzzing with their own chatter. Each one is a glowing sphere or spiky bubble, shimmering with colorless light, humming in patterns that you never consciously notice. They're talking right now, chemical murmurs, electrical sparks, protein handshakes, and whether you feel ready for it or not, you are completely at the mercy of this invisible orchestra. You probably won't survive this without them. So, before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. And while you're at it, let me know in the comments where you're tuning in from and what time it is for you. It's oddly grounding to realize that while your cells are whispering in biochemical code, we humans are sending little messages to each other across time zones. Now dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. The first thing you notice is the music. Not literal sound, of course. No tiny cell is blasting a playlist of whale songs, but waves of chemical signals rising and falling like notes. You drift through the bloodstream, that velvet dark river, and see red blood cells stacked like coin towers, sliding past with the ease of inner-city taxis. They don't have nuclei, which is a fun biological fact. They've ditched their DNA libraries to carry more oxygen instead, like minimalist monks who threw out all their furniture so they could move faster. Historians still argue whether the discovery of red blood cells in the 17th century by Jan Swammerdam or Antony van Leeuwenhoek deserves more credit. Either way, those early microscopists were basically the first paparazzi of the cellular world. As you float closer, the symphony sharpens. Each cell pulses faintly, glowing with ions flickering like Morse code. Some messages are urgent. Calcium sparks summoning muscle fibers to contract, sodium waves crackling across neurons like fireworks. Others are quieter, slower, hormones floating lazily down the bloodstream like love letters tossed in bottles. You begin to realize that survival isn't a single drumbeat. It's an overlapping choir, a messy jam session, sometimes more jazz than symphony. Occasionally, the chatter goes wrong. A misfolded protein stumbles onto the stage like someone who misread the invite and showed up to a masquerade ball in sweatpants. The surrounding proteins gently escorted away, like embarrassed bouncers. Sometimes the system fails, though, and those misfolded proteins pile up, forming plaques linked to diseases like Alzheimer's. It's unsettling, like discovering the gossip circle in your neighborhood can, if left unchecked, turn into a mob. But right now, in this drifting, dreamlike state, you're noticing the elegance. The cell membranes shimmer as boundaries, semi-permeable doors opening and closing, letting some molecules in while keeping others out. It's like a VIP club, but instead of bass thumping and overpriced cocktails, the entry requirement is simply whether your molecular shape fits the lock. A glucose molecule floats up like someone flashing the right wristband, and the gate swings open. Meanwhile, a stray bacterium tries to sneak in uninvited and immediately gets tackled by a white blood cell that's basically been waiting its whole life for this dramatic takedown. There's humor even here in the strange seriousness of your cellular citizens. Imagine if you could translate their conversations into English. "Hey, potassium, can you leave the cell for a sec? Thanks, we're doing a nerve impulse." Or, "Attention, mitochondria, we need extra ATP stat. No, I don't care if you're tired." You discover your body runs on the kind of chaotic group chat energy where everyone is talking over each other, but somehow, against all odds, things get done. What's quirky is that some animals literally do glow from cellular chatter. Fireflies, for example, coordinate their lights with a chemical signal called luciferin, an actual glow word like a neon emoji. Your own cells aren't bioluminescent, but the principle is the same. Chemistry becoming communication, signal becoming spectacle. And here's the weird part. Scientists still argue whether cells intend anything at all, or if these signals are just automatic reflexes without meaning. It's like debating whether your phone is actually talking to the Wi-Fi router, or just blindly pinging. As you float along, your sense of scale collapses. 1 second you feel as small as a virus. The next you're overwhelmed by the idea that this entire buzzing metropolis exists inside you every second without asking permission. The intimacy is almost embarrassing, like catching your roommate rehearsing a secret dance routine in their bedroom. You never thought to ask your cells what they were up to. Yet here they are, keeping you alive with a language so complex that even the sharpest scientists still struggle to parse its grammar. The bloodstream carries you deeper and the rhythms grow denser. The symphony isn't uniform. Some tissues hum with rapid chatter, like neurons in the brain firing in high-speed bursts. Others are slow, steady drummers, liver cells detoxing, skin cells patching up scrapes you forgot you had. Each has its dialect, its slang, its inside jokes. If you could eavesdrop long enough, maybe you'd find that your own personality isn't just you, but the sum of these molecular conversations, endlessly negotiating in the dark. You drift further, lulled by the overlapping signals, until the chatter feels less like noise and more like a lullaby. The cells aren't frantic, they're just alive. They hum the way stars burn, the way tides shift, the way life insists on persisting message by message, spark by spark. You ease forward, carried deeper into the glowing labyrinth, and your attention lingers on a particular boundary, the surface of a single cell. From a distance, it looks like a soap bubble floating in the dark, but as you move closer, you notice it's anything but flimsy. The cell membrane undulates, alive, shimmering like a velvet curtain woven from living molecules. It's not just a wall, it's a nightclub bouncer, a translator at the UN, and your building's doorman, all rolled into one. Nothing gets through without permission. The structure is elegant, a bilayer of phospholipids, each with a head that loves water and a tail that hates it, stacked in perfect alignment. This weird arrangement means the cell can seal itself off while still interacting with the world outside. You hover at its edge and see proteins embedded like door handles, rotating, shifting, opening, closing. Some of them are channels, gated like subway turnstiles, letting sodium, potassium, or chloride ions through. Others are receptors, little antennas twitching for signals. Without this constant negotiation, you'd be a puddle of chemistry instead of a coherent body. There's history here. Back in the 1920s, scientists Gorder and Grendel proposed that membranes are made of a lipid bilayer. The way they figured it out, they squashed red blood cells, spread out their lipids on water, and realized the area doubled what one layer would cover. A messy but oddly poetic experiment, like pressing a flower between book pages, and realizing it explained the entire concept of cellular walls. Historians still argue whether their math was completely solid, but the idea stuck, and now it's in every biology textbook. As you watch, a hormone molecule drifts near, a tiny courier with a message. It docks into a receptor, and instantly the cell membrane shudders with activity. A cascade of signals ripple inward, like gossip spreading through a crowded party. The outer surface is only the start. It's a switchboard, passing along whispers to the cell's interior machinery. The receptor itself is like that one friend who hears a rumor and immediately texts 5 different group chats. And then, a funny thought hits you. If your body's cells are constantly talking at this microscopic level, what if some of them are chronic oversharers? Maybe a liver cell is the office gossip, forwarding every chemical headline. Maybe your neurons are the poets, exaggerating with electric flair. And maybe your skin cells are the quiet ones at the back of the classroom, dutifully holding the boundary and pretending they don't hear the drama. You drift sideways and brush against the membrane itself. To your surprise, it isn't smooth. It's covered in tiny sugar chains like frosted sprinkles on a donut. This sugary coat, the glycocalyx, helps cells recognize each other, the same way name tags help strangers at a conference. It's endearing and a little silly. Imagine your cells constantly checking name tags. Hello, fellow kidney cell, nice glycocalyx you've got there. The quirky part is that some pathogens, like viruses, sneak in by mimicking these sugary decorations, tricking the bouncer into letting them through. The original social engineering hack. The cell surface also crackles with voltage differences. Inside is slightly negative, outside slightly positive. That polarity keeps the gates twitching, ready to spring open. It's like the electrical tension in a theater before the curtains rise, audience murmuring, stage lights hot, everyone waiting for the cue. And yet this shimmering, whispering membrane isn't invincible. Too much heat, too little oxygen, or a careless toxin can punch holes in it, and the symphony falters. When that happens, cells burst like soap bubbles, and the neighboring tissues must clean up the mess. Some scholars even debate whether the membrane is the true seat of the cell's intelligence, since it's here that choices about what gets in and out are made. Is the nucleus really the boss, or just a librarian following requests from the gatekeepers? While you're drifting, a bizarre historical tidbit surfaces in your mind. In the 1930s, some scientists believed membranes were coated in crystalline proteins arranged like tiles in a mosaic bathroom floor. The model turned out to be half right, half imagined, but the image stuck, and today we still describe it as the fluid mosaic model. It's not entirely precise, but let's be honest. Fluid mosaic does sound a lot prettier than jiggly lipid soup with embedded doodads. Here, close up, the beauty feels undeniable. The lipid tails sway like seaweed, the proteins bob and shift, the sugars sparkle faintly. It looks alive because it is alive. And you, floating just outside the wall, realize you've been staring at the front door of your own survival this entire time. The cell isn't sealed in solitude. It's tuned into every passing molecule, every chemical breeze, every whisper of calcium or dopamine. Without this gatekeeping conversation, you'd never have taken your first breath. You catch one more sight before drifting away. A white blood cell bumps against the membrane, sniffing it suspiciously. After a moment, it moves on. No enemy here. For now, this cell is safe, its whispers intact. But the thought lingers. Your survival depends on billions of these tiny boundaries making good choices, second after second, without ever asking your permission. You drift along the surface of that shimmering membrane, watching the little sugar sprinkles and embedded proteins sway, until something catches your eye. A molecule, drifting lazily in the bloodstream, suddenly snaps into place on the membrane. It's like watching a dancer step perfectly onto a partner's hand, the choreography so precise it feels rehearsed for eons. This is a receptor ballet, and you've just snagged front row tickets. The docking is specific, painfully specific. The receptor only accepts one shape, like a lock waiting for the right key. You imagine a frustrated molecule showing up in the wrong outfit. Sorry, you're not on the list. Please try another cell. The successful match, though, sparks a ripple. Proteins inside the cell shift, enzymes activate, and the membrane whispers its new instructions inward. The entire cell listens, as if the outside world just handed it a folded note in class. Historically, the fascination with receptors really took off in the early 20th century. Paul Ehrlich, a German scientist, famously described them as side chains on cells that could bind with toxins or drugs. He used a vivid metaphor, keys and locks, which stuck. Historians still argue whether his magic bullet idea of targeting specific receptors oversimplified biology or actually revolutionized it. Either way, thanks to Ehrlich's obsession, we now design medicines that fit these locks with uncanny precision. And that's where it gets fun. Think of how caffeine keeps you awake. It doesn't give you energy directly. It just docks onto receptors meant for adenosine, a molecule that tells your brain, hey, you're tired. Caffeine sits there like an imposter, blocking the message, and suddenly your neurons keep firing without realizing they're exhausted. It's the cellular equivalent of putting tape over your car. Low fuel, light, and pretending you're fine. Inside the cell, the signal branches out in cascades. You see proteins passing the message like relay runners, each one flipping into a new shape as they sprint forward. This shape-shifting is mesmerizing. Proteins don't just sit still. They fold and unfold, twist and wiggle, like little origami artists flipping themselves inside out. Sometimes they carry phosphate groups as if handing off batons, sparking the next step in the chain. It's drama, really, soap opera levels of gossip. Kinase says we're turning on gene expression. No way, I heard phosphatase just shut it down. Here's a quirky side note. Scientists once found receptors for morphine in the brain before they figured out the body makes its own morphine-like molecules, called endorphins. It was like finding a keyhole in your house that didn't match any of your keys, and then realizing your body's been manufacturing secret keys all along. You almost want to laugh. Your cells were keeping a stash of natural painkillers and just never told you. The ballet isn't always graceful, though. Sometimes the wrong molecule sneaks in and triggers chaos. Certain snake venoms, for example, hijack receptors, jamming them open or shut. One toxin from the banded kite snake blocks communication at the neuromuscular junction, effectively cutting off the body's ability to move. It's terrifying, but also oddly cinematic. The villain doesn't need to destroy the city. Just hack into the traffic lights and watch the gridlock. Watching the receptors in action, you get the sense that no single molecule matters. It's the choreography that counts. A cell might be getting thousands of signals at once, some urgent, some routine, some contradicting each other. The receptors are like doormen at a hotel where 5 different conventions are happening simultaneously. One for wedding planners, one for dentists, one for extreme unicyclists, and one for UFO enthusiasts. Somehow, the doormen keep track of who belongs where, and the hotel doesn't collapse. Scholars still debate just how smart receptors are. Are they mere mechanical parts, opening and closing without thought? Or do they represent a kind of primitive information processing, a cellular sense of awareness? The argument echoes bigger questions. At what point does chemistry turn into communication? And when does communication become consciousness? It's one of those puzzles you'll still be turning over in your head as you drift towards sleep. As you drift closer, you notice how receptors cluster. They don't stand alone like lonely statues. They huddle in rafts, little molecular cliques hanging out together. When one activates, others join in, amplifying the signal. You can't help but imagine them as gossipy friends at a coffee shop. One whispers something scandalous, and suddenly the entire table is leaning in, wide-eyed, deciding to take action. And yes, sometimes the gossip is true. Sometimes it's overblown, but either way it spreads. The longer you watch, the more beautiful it seems. These microscopic dances are happening right now in your own body. Every thought you have, every blink of your eye, every dream you're drifting into as you listen. It's all being choreographed by receptors passing messages in elegant, invisible ballet. The quiet realization sinks in. You are not just living in your body. You are the sum of these dances, millions upon millions of tiny locks and keys opening, closing, and whispering secrets you'll never fully hear. The ballet fades, and now your gaze falls on a doorway, a protein channel embedded in the membrane. It looks like a tunnel opening, a small ring of shifting molecules, and for a moment nothing happens. Then suddenly ions pour through. Sodium and potassium, tiny charged beads of matter, rushing like commuters through a turnstile. You can almost hear them clicking, the turnstile whirring, each ion carrying a fragment of electric potential into or out of the cell. This is the true heartbeat of communication, ions at the gates. The flow isn't random. It's carefully timed, choreographed, and pulsed in waves. In your neurons, sodium charges flood in, potassium charges flush out, and that tiny imbalance sparks a nerve impulse. That impulse races down your spinal cord and before you even notice, tells your hand to twitch, your lips to form a word, or your eyelids to droop just slightly heavier. Your body is basically powered by controlled leaks. Historically, one of the great leaps in understanding this came from the work of Alan Hodgkin and Andrew Huxley in the 1950s. They used giant squid axons, seriously, squid the size of your living room, to measure the flow of ions and model how nerve impulses propagate. They even won a Nobel Prize for it. Historians still argue whether their models were overly simplified, but those squid experiments unlocked the electrical code of life. Without them, you might not have half the medical technology we take for granted today. You notice the floodgates opening again, this time calcium ions slipping through. They're not here for electricity, they're here to signal. In muscle cells, calcium pours out of storage compartments, telling fibers to contract. Without it, your heart wouldn't beat, your fingers wouldn't flex, and your best dance move would remain theoretical. Calcium is also a trigger for cell death, a paradoxical role, like the fire alarm that both saves the building and, if too persistent, tells everyone to demolish it. There's a quirky detail too. In the 19th century, some early physiologists believed nerve signals were literally animal electricity, like tiny lightning bolts crackling inside you. In a sense, they weren't entirely wrong, but they pictured it more like thunderbolts instead of controlled ion shifts. Imagine if your every thought came with a spark shooting out of your ears. You'd need flame-retardant pillows just to survive REM sleep. Back inside the cell, you see how this ionic flow shapes everything. Chloride ions balance charges, potassium stabilizes the resting potential, sodium drives nutrient transport. They're like a trio of old musicians who have been playing together forever. No showy solos, just the constant background groove that makes the whole band work. And when the rhythm falters, say, too little potassium from dehydration, the whole performance wobbles. That's why even mild electrolyte imbalance can leave you dizzy, weak, or staring blankly at your fridge wondering why you came into the kitchen. The gates themselves are fascinating. Some respond to voltage changes, open when the inside gets too negative, close when it balances out. Others respond to molecules binding, like neurotransmitters knocking politely before the door swings wide. Still others respond to physical stretch, like channels in your ear cells that open when sound waves jiggle them. Your entire sensory world, every song you hear, every taste of chocolate, every itchy sweater, is built on these molecular gates flicking open and shut. Scholars still debate just how synchronized these gates really are. Do they open independently, like popcorn kernels in a pan? Or do they coordinate like soldiers marching in lockstep? Some experiments suggest bursts of cooperative behavior, little flurries of ions charging together. If true, it means even at this microscopic level, your body prefers teamwork over pure chaos, though the teamwork sometimes looks suspiciously like a flash mob. You catch a moment of mischief. A toxin from a pufferfish molecule sneaks into the channel, wedging it shut. This is tetrodotoxin, one of nature's nastiest tricks. It paralyzes by blocking sodium channels, silencing the flow of messages in your nerves. To you, drifting safely in imagination, it's only eerie. To real cells, it's a death sentence, a forced stillness in the symphony of charges. your respect for ion gates deepens. They're fragile, yet somehow they keep you moving, thinking and dreaming night after night. What strikes you most is how little space these ions travel, just across the width of a membrane, mere nanometers. Yet those tiny hops scale upward into thoughts, songs, entire civilizations. The smallest sparks of charge become the great arcs of human history. Imagine your decision to press play on this video, the flicker of curiosity that brought you here, was a ripple of sodium and potassium gates firing open in your cortex. Your cells didn't ask, they just pushed the ions and you followed. You drift closer, peering through the tunnel of a single channel. Inside, the ions aren't just rushing blindly. They're guided by charges, tugged along by fields invisible to your eye. It's like watching an airport conveyor belt that always brings you exactly where you need to go. The elegance of it feels almost unfair. You've been struggling to remember where you left your keys, and meanwhile your cells are running full-scale electrochemical highways without ever missing a beat. The realization settles like a soft blanket. Every flicker of awareness you feel depends on these gates behaving themselves. Your cells are fluent in the language of ions, and you're just along for the ride, mistaking their music for your own thoughts. The ion gates quiet behind you, fatting like the afterecho of distant drums, and you find yourself drifting into the spacious interior of a single cell. It feels like stepping into a cathedral, vast, shimmering, filled with echoes. This is the cytoplasm, the jelly-like matrix where everything floats, collides, and colludes. At first it looks still, but as your eyes adjust, you notice the bustle. Vesicles scooting along on microscopic tracks, enzymes tumbling, organelles bobbing like carnival balloons. It's gossip central, but instead of words, the chatter happens in molecules. The cytoplasm isn't just goo. It's a crowded marketplace where proteins trade information, ions pass notes, and organelles shout across the room. Imagine Times Square on New Year's Eve, except every confetti piece is a molecule carrying a message. Your neurons, for example, rely on calcium signals spreading through the cytoplasm like a wave of applause. A little burst near the membrane sparks a bigger ripple toward the nucleus, and suddenly, genes are turning on or off. It's messy but strangely coordinated, like an orchestra without a conductor, yet somehow everyone knows when to come in. Historically, scientists underestimated this space. In the 19th century, cytoplasm was dismissed as protoplasm, a boring filler like packing peanuts around the important stuff. But then discoveries started piling in, molecular motors, signaling cascades, cytoskeletal highways, and suddenly it wasn't filler anymore. It was the stage itself. Historians still argue whether the shift from protoplasm to cytoplasm marked a revolution in cell theory or just a semantic upgrade. Either way, the goo had its glow-up. Your gaze catches one of those molecular motors now. A chitin protein, shaped like a little stick figure with two legs, is literally walking along a microtubule, dragging a vesicle behind it like a weary parent pulling a suitcase through an airport. Step by step, powered by ATP, it trudges across the cytoplasmic landscape. It's both hilarious and awe-inspiring. Some researchers even animate them for fun, turning these tiny commuters into stars of viral YouTube videos. And here it is, live in your body right now, hauling its precious cargo. Nearby, ribosomes dot the endoplasmic reticulum, busy factories churning out proteins. Each one receives an RNA transcript and translates it, clicking amino acids into place like a line cook flipping pancakes. The cytoplasm hums with their activity, protein gossip spreading from one corner to another. One ribosome says, "We're making insulin today," and before you know it, a cascade of molecules is adjusting blood sugar. Another says, "We're assembling antibodies," and suddenly your immune system's war room lights up. The chatter never ends. Here's a quirky tidbit. Some biologists describe cytoplasmic signaling as noise. Not noise in the annoying sense, but in the statistical sense, random fluctuations that sometimes create new, surprising behaviors. Think of it as cellular jazz improvisation. One cell might interpret the noise as a signal to divide, another as a reason to wait. Historians of science still debate whether this noise is a bug in the system or an essential feature of life's adaptability. After all, too much order is boring. A little chaos keeps things interesting. And speaking of chaos, you spot a lysosome, basically the cell's trash can scooping up debris. Enzymes inside chew through broken proteins and worn-out organelles. It's a recycling program, but you can't help picturing it as a gossipy neighbor rooting through everyone's garbage. Oh look, another misfolded protein. Tusk. Without lysosomes, though, the cytoplasm would drown in its own clutter. It's weirdly comforting to know your cells are tidier than your closet. The atmosphere feels alive but not frantic. Molecules diffuse lazily, bouncing like beach balls in a crowded pool. Signals dart from one organelle to another, sometimes colliding, sometimes fizzling out. The overall effect is like background gossip at a cafe, lively, constant, but not overwhelming. You imagine each whisper as a sentence. Tell the mitochondria we need more ATP. Inform the nucleus it's time to divide. Remind the membrane to keep out that sketchy virus. The cytoplasm is essentially your body's group chat, with messages flying at all hours, sometimes coherent, sometimes chaotic, but always vital. There's also beauty in how temporary everything feels. A messenger molecule appears, drifts across the cytoplasm, delivers its news, and then dissolves. Nothing lingers too long. It's like a dream you remember in the morning, important in the moment, gone by sunrise. And yet somehow this constant turnover sustains you. The gossip never stops, but neither does it overwhelm. It's the steady hum of aliveness. You can't help but chuckle at a thought. If your cells had a rumor mill, would it ever misfire? Maybe one vesicle whispers, "Divide now," but the lysosome overhears it and says, "Did you hear? They're all dividing tomorrow." Suddenly, half the cell is preparing for mitosis while the other half is still making dinner. In truth, miscommunications like that do happen, and when they spiral, you get cancer or autoimmune disease. Even at this scale, gossip can ruin lives. Still, watching the cytoplasmic swirl, you feel a strange calm. It's chaotic, yes, but it's also endlessly resourceful. Signals bounce, molecules march, vesicles ferry their loads, and somehow it all adds up to a coherent you. The chatter isn't noise, it's a lullaby, one you've been listening to since the moment your first cells formed. You drift deeper into the cell's interior, leaving behind the noisy, bustling cytoplasmic bazaar. Ahead in the center looms something more reserved, the nucleus. Compared to the constant chatter of vesicles and proteins outside, this place feels hushed, like stepping into a library at midnight. The membrane surrounding it, double-layered and studded with pores, looks almost ceremonial, a velvet rope separating the dance floor from the reading room. And that's fitting, because the nucleus isn't here to party. It's the introverted librarian of the cell, keeping all the DNA manuscripts under careful watch. Inside, you see the chromatin, long threads of DNA wrapped around histone proteins like spools of yarn. They're not spread out evenly. Some regions are tightly packed, silent, while others are loose and fluttering, ready to be read. You float closer, watching as enzymes move along the strands, unzipping sections, copying out instructions. It's quieter than the cytoplasm, but not lifeless. Every turn of the helix, every gentle tug of a polymerase enzyme is another page being photocopied. Historically, the nucleus has always carried an air of mystery. In the 1830s, Scottish botanist Robert Brown was one of the first to describe it formally, peering through his microscope and spotting the little dark spot inside orchid cells. Historians still argue whether Brown truly understood its importance or just wrote down what he saw. Either way, his observations kicked off 2 centuries of fascination, and the nucleus became biology's equivalent of the Vatican vault, sacred, secret, and full of critical texts. You hover near one of the nuclear pores, vast protein complexes that look like ornate archways. Molecules line up politely to pass through. An mRNA transcript, newly copied, slips out of the nucleus like a note being smuggled past a stern librarian. Proteins float in, sometimes carrying permission slips, sometimes getting turned away. It's strangely bureaucratic. You half expect someone to stamp approved in red ink on each molecule before it passes through. And yet the librarian analogy fits. The DNA inside is not shouting orders. It doesn't blast announcements across the cytoplasm. Instead, it releases instructions only when asked. A signal arrives at the membrane, cascades through the cytoplasm, taps politely at the nuclear door, and only then does the DNA uncurl and share a recipe. Quiet, deliberate, almost shy. If the cytoplasm is a crowded cafe, the nucleus is a single reader at the back table, carefully copying notes into a leather-bound journal. There's a quirky tidbit here. Some DNA is never used at all. Vast stretches of your genome sit in silence, once dismissed as junk DNA. Today, scientists know some of it plays regulatory roles, but much remains enigmatic. Is it truly garbage, or is it poetry you just don't know how to read yet? Scholars still debate the point, and every year brings new surprises. Imagine the librarian insisting, "Don't touch that dusty shelf. It might look like gibberish, but someday it could save your life." As you float between the chromatin threads, you're struck by how impossibly long they are. If you unspooled all the DNA from just one of your cells, it would stretch nearly 2 meters. Yet it's packed so neatly, so elegantly, that it all fits inside a structure barely a few micrometers wide. It's the ultimate space-saving trick, better than any vacuum-sealed storage bag. You feel small inside this labyrinth of instructions, like wandering through a manuscript written across miles of parchment. You notice that the DNA isn't static. It coils and uncoils, responds to signals, even loops to bring distant sections into contact. Some scientists describe this as a kind of 3D choreography, where the genome folds itself into specific shapes to control what gets expressed. You can't help but picture the DNA as a shy but dramatic stage performer, arranging props in just the right positions before delivering a soliloquy. Occasionally, though, mistakes happen. A polymerase enzyme slips, a base pair gets miscopied, and the librarian frowns. Most of the time, repair crews rush in, enzymes patching the error, proofreading the script. But if too many mistakes slip past, the cell risks malfunction, even cancer. It's a sobering reminder that even the quietest libraries are vulnerable to gossip, graffiti, and the occasional fire. You chuckle at another thought. What if the nucleus had a personality? It'd probably shush everyone constantly. The mitochondria would be too loud with their power-making drama, the ribosomes too messy with their protein clatter. Meanwhile, the nucleus would mutter, "Keep it down. I'm trying to copy instructions for hemoglobin over here." And yet, for all its introversion, the cell can't function without it. The librarian may be quiet, but without those manuscripts, the party outside would collapse into silence. You take one last look around the hushed chamber. DNA threads glimmer faintly, proteins shuffle in respectful silence, pores stand guard. It feels solemn, timeless, and oddly comforting. You realize you're floating inside the blueprint of yourself. Every eye color, every habit, every quirk written here in molecular ink. The nucleus doesn't boast, doesn't command attention, but in its quiet, steady way, it carries your story. You slip out of the nucleus, past the quiet guardians at the pores, and find yourself trailing one of those freshly copied messages. It's an mRNA strand, shimmering like a ribbon of light, curling slightly as it floats through the cytoplasm. This is RNA, the sassy middle manager of the cellular world. DNA might be the ultimate authority, carefully guarded in the nucleus, but it rarely leaves its office. Instead, it sends RNA out to handle the day-to-day business, carrying instructions, making edits, and sometimes, just sometimes, freelancing its own ideas. The strand you follow wiggles with purpose like a courier late for work. Its bases, adenine, cytosine, guanine, and uracil, form the coded message, 3 letters at a time, spelling out which amino acids should be strung into a protein. You can almost hear it muttering, "Yes, yes, DNA says we're making hemoglobin today. Don't argue, just build." The ribosomes are waiting, coffee in hand, ready to translate. Historically, the discovery of RNA's role was a huge deal. In the 1950s and 60s, the central dogma of molecular biology, DNA makes RNA makes protein, was proposed. But even then, RNA kept surprising people. Historians still argue whether the term dogma was an overstatement, since RNA is not just a messenger. It sometimes edits DNA, silences genes, or even carries out catalytic jobs of its own. The story keeps getting messier, like a middle manager who not only delivers memos, but also rearranges the office furniture without telling anyone. You drift closer as the mRNA gets clamped into a ribosome. The code is read 3 bases at a time, triplets called codons, and each codon calls for a specific amino acid. Transfer RNAs, those quirky little L-shaped molecules, swoop in with amino acids attached like tiny gifts. They line up at the ribosome, offering their packages in the correct order. It's a little like a sushi conveyor belt, except every roll has to be placed precisely to make a working protein. Miss a codon, and the whole dish comes out wrong. And here's a quirky tidbit. Some viruses are basically just RNA wrapped in protein. The flu, measles, and coronaviruses, all RNA genomes that hijack your cells. They don't bother with their own factories. They just waltz in and hand your ribosomes counterfeit memos. Surprise, you're making spike proteins now. The ribosomes, ever dutiful, follow the instructions without complaint. It's the ultimate workplace prank, except the prankster can knock you flat in bed for a week. Not all RNA is bossy, though. Some RNAs float around as regulators, silencing certain genes, or even splicing themselves into new shapes. One type, microRNA, is so tiny it was overlooked for decades, yet it plays a huge role in controlling which proteins get made. Scientists only really started paying attention in the 1990s, which in biology terms is practically yesterday. Even now, scholars debate just how much control microRNAs exert. Are they minor editors? Or do they secretly run the whole newsroom? You follow another RNA strand, this one editing itself as it goes. It snips out introns, nonsense sequences that don't code for proteins, and stitches together exons, the meaningful bits. The editing is so elaborate it feels like performance art. Some genes can be spliced in multiple ways, creating entirely different proteins from the same stretch of DNA. It's like a choose-your-own-adventure book where one version ends in a pirate battle and another in quiet tea by the fire. The flexibility is staggering. You can't help but smile at the attitude of RNA. DNA is stable, double-stranded, tucked safely in the nucleus like a serious professor. RNA is single-stranded, fragile, fleeting, and constantly improvising. It shows up, makes things happen, and dissolves in hours or days. You imagine it wearing flashy sneakers and carrying a latte, rolling its eyes at the old librarian back in the nucleus. "Relax, I've got this," it says, before promptly getting degraded by an enzyme. Classic middle manager, loud, effective, and occasionally disposable. But here, where it gets wild, some scientists suspect that before DNA, before proteins, The earliest life on Earth was run by RNA alone. They call it the RNA world hypothesis. In this view, ancient RNA molecules both stored genetic information and catalyzed chemical reactions, pulling double duty like overworked interns. If true, it means your sassy little messenger molecules are echoes of life's original language. Scholars still argue how plausible this is, but the thought lingers. Maybe RNA isn't just a middle manager. Maybe it's the founder who stuck around, still running errands while everyone else pretends they're in charge. The ribosome hums, amino acids click into place, and the protein begins to fold. The mRNA mission accomplished drifts off, already starting to fray. Its lifespan is short, but its influence is immense. Watching it dissolve, you realize how much of your existence depends on these fleeting strands. Every breath, every blink, every dream, carried on the backs of molecules that live only long enough to pass the message. The protein emerges from the ribosome like a ribbon spilling from a spool. At first, it's just a simple chain of amino acids, beads strung together one by one. But almost immediately, the ribbon begins to twist, fold, and buckle in on itself. Loops form, spirals coil, sheets press flat. It's like watching someone fold a piece of origami at lightning speed, except instead of cranes or frogs, the end result is a three-dimensional molecular machine. This is the chatter of proteins, each one shaping itself into a voice in the cellular conversation. Proteins are everywhere. They're the enzymes digesting your dinner, the collagen scaffolding your skin, the antibodies patrolling your bloodstream. Some are stiff and structural, like girders in a building. Others are flexible, darting around to deliver messages. They're not just parts, they're workers, gossipers, and occasionally troublemakers. A single protein might spend its life ferrying cargo, snipping bonds, or plugging into a receptor like a USB drive. Multiply that by tens of thousands, and you've got the humming, talkative workforce that keeps you alive. Historically, proteins were once thought of simply as chains of stuff. It wasn't until the mid-20th century that their folded shapes were revealed, thanks to techniques like X-ray crystallography. Dorothy Hodgkin, for example, cracked the structures of insulin and vitamin B12, laboring for years over data that today's computers would solve in minutes. Historians still argue whether her meticulous patience or the leap of computational modeling truly advanced the field more. Either way, those early efforts uncovered the secret. Form equals function. Without the right fold, the chatter stops. You hover close to a newly made protein still folding. Its amino acids tug on each other, some repelling water, others seeking it, like introverts and extroverts arranging themselves at a party. The process looks chaotic, but somehow in milliseconds, the ribbon snaps into its final shape. You can't help but grin. It's like watching someone solve a Rubik's Cube blindfolded, faster than you could line up even two colors. Of course, folding doesn't always go right. Misfolded proteins can clump together, forming sticky aggregates. In your brain, that can lead to conditions like Alzheimer's or Parkinson's. You imagine a protein showing up at a cocktail party in the wrong outfit, then stubbornly refusing to leave, gathering friends until the dance floor is blocked. Quirky side fact: in cows, a misfolded protein called a prion can trigger mad cow disease. Unlike other pathogens, prions have no DNA or RNA. They're just proteins gone rogue, teaching others to fold badly. It's like a bad fashion trend spreading through your wardrobe until suddenly all your socks are wearing sunglasses indoors. Despite the risks, proteins are dazzlingly versatile. Some act like keys, unlocking reactions by lowering energy barriers. Others are literal machines. ATP synthase, for example, spins like a turbine, cranking out cellular fuel. Watching it whirl, you feel like you've stumbled into a microscopic factory where every worker knows their role. And yet, the chatter never stops. Proteins nudge each other, bump into receptors, form complexes, break apart again. The cell is a cocktail party where everyone has a task but still manages to mingle. You spot a hemoglobin protein drifting by, shaped like a cluster of cushions. It opens and closes to carry oxygen, releasing it where tissues need it most. Without this protein chatter, your body's delivery system would collapse. A fun historical tidbit: hemoglobin was the very first protein structure solved by X-ray crystallography in the 1950s. Scientists were so obsessed with it that one famously described it as the molecule that made us human. Scholars still debate whether that's poetic exaggeration or a legitimate claim. There's also flair in the diversity of proteins. Antibodies look like little Ys, waving their arms around, searching for intruders. Actin filaments assemble into highways, while motor proteins march along them like postal workers. Even insulin, a humble hormone, folds itself with just enough elegance to dock neatly with its receptor. Each protein is a character in the drama, some bold, some quiet, some tragicomic in their dysfunction. As you drift among them, you realize proteins aren't just talking to each other. They're talking to you. Every emotion you've ever felt, joy, fear, boredom, is tied to proteins moving ions, folding receptors, sparking cascades. Every laugh you've had came from proteins nudging neurons to fire. Every dream you're slipping into now is sculpted by their chatter. You're the sum of their conversation, even though you never hear it directly. And still, scholars debate the boundaries. At what point does protein chatter become meaning? Is an enzyme folding sugar molecules into glycogen just chemistry, or is it communication? Some insist it's only mechanics. Others argue that life itself is the meaning that arises from the chatter. You find the debate oddly soothing, because whether you call it noise or language, the proteins keep folding, whispering, and building you, night after night. One last protein catches your eye: chaperones. These aren't structural or catalytic, they're caretakers, hovering nearby to help other proteins fold correctly. If something starts to misfold, a chaperone steps in, coaxing it back into place. You can't help but laugh. Molecular babysitters making sure the toddlers of amino acids don't put their heads through the armholes of their shirts. Without them, chaos would reign. With them, the chatter becomes symphony again. You drift past the folding frenzy of proteins and catch sight of something glowing with a strange inner fire. At first you think it's just another organelle, but as you approach, you recognize the shape. A bean-like capsule with inner membranes looping back and forth like a folded accordion. This is a mitochondrion, the powerhouse, the drama queen, and the adopted child of the cell. Inside, the air hums with energy. Molecules of glucose, broken down earlier, arrive like delivery trucks. Enzymes dismantle them, stripping electrons, passing them along chains of proteins embedded in the inner membrane. With each handoff, protons are pumped across the membrane, building a gradient, a chemical dam straining with potential. Then comes the turbine, ATP synthase, a molecular machine that spins as protons rush back through, minting fresh ATP molecules with every turn. ATP, your universal energy coin. Without it, you wouldn't take another step, another breath, another thought. The mitochondria are printing money 24 hours a day, and your entire economy depends on them. The drama, of course, comes from their history. Mitochondria weren't always part of you. Billions of years ago, they were free-living bacteria, swallowed by larger cells. Instead of being digested, they struck a deal. We'll make you energy if you let us stay. That symbiosis was so successful it became permanent. Every one of your cells still carries them around, like roommates who never moved out. Historians still argue whether it was cooperation or conquest, whether the larger cell invited the bacteria in or enslaved it. Either way, you're the product of that uneasy alliance. Quirky fact: mitochondria still carry their own DNA, a remnant of their independence. It's tiny compared to your nuclear DNA, but crucial. Mutations here can cause diseases ranging from muscle weakness to neurological problems. Even stranger, mitochondrial DNA is inherited almost exclusively from your mother. It's like a matrilineal family name written into your cells, passed down without interruption. Every mitochondrion in your body is a direct heirloom from your maternal ancestors, stretching back through time. You drift closer, watching the inner membrane folds, the cristae, where the electron transport chain hums like a conveyor belt. The place is buzzing, yet oddly claustrophobic, as if the walls themselves are charged with potential. The hum intensifies, and you realize this is where your very warmth comes from. A little inefficiency in the system releases heat, and without it, you'd be a cold, lifeless figure. Even the cozy feeling of blankets at night is secretly powered by these inner sparks. Mitochondria aren't just engines, they're also decision makers. When a cell is too damaged to survive, mitochondria release cytochrome C, a signal that triggers apoptosis, the elegant cell death program. It's chilling, like discovering your roommate isn't just making dinner but also has the power to evict you permanently. Life and death, both negotiated in these bean-shaped factories. You think of the mitochondria as theatrical, always central to the story, always humming with intensity. They even get blamed for aging. Some researchers believe damage from reactive oxygen species, byproducts of all that energy production, gradually wears down cells, leading to wrinkles, gray hair, and creaky joints. Scholars still debate whether this free radical theory of aging is the main culprit or just one piece of a bigger puzzle. Either way, mitochondria remain prime suspects in the case of why we don't stay young forever. Here's something quirky and oddly endearing. Some people half-jokingly call mitochondria the bacterial in-laws of your body. They weren't invited. They don't always play nice, but you can't function without them. And like family gatherings, their drama sometimes spills out. Mutations, malfunctions, and power struggles. Yet their contributions are undeniable. They built the energy surplus that allowed multicellular life to flourish. Without their deal, Earth might still be covered only in single-celled wanderers. You hover above a cluster of mitochondria, noticing they're not isolated at all. They fuse, divide, and network, constantly reshaping themselves. In neurons especially, they line up along axons like street lamps, powering the electrical impulses that race from brain to body. You picture them as a chain of tiny nightlights, keeping the highway lit. Without them, your thoughts would fizzle in darkness. The more you watch, the more you realize, mitochondria aren't just factories. They're storytellers, weaving ancient bacterial survival strategies into the fabric of your being. Their hum isn't just chemistry, it's a reminder of the oldest gossip in biology, whispered across billions of years. Let's stick together. It's the only way to survive. The mitochondrial hum fades behind you, replaced by sharper, quicker flashes of light. At first, you think it's random static, but then you recognize the pattern. Calcium ions, darting in and out of compartments, like sparks from a hidden fireworks show. These tiny bursts are secret handshakes, the silent signals that cells use to control some of their most important moves. A calcium ion slips through a channel in the endoplasmic reticulum, and instantly the cytoplasm brightens in a ripple, like a stone dropped into a pond. Proteins snap to attention. Muscle fibers contract. Vesicles release their cargo. Enzymes activate. Calcium is the exclamation point in the cell's language. One brief influx, and an entire system responds. Historically, calcium signaling was puzzling. In the 19th century, physiologists knew muscles needed calcium salts to contract, but they had no clue how. It wasn't until the mid-20th century that researchers uncovered the sarcoplasmic reticulum, the muscle cell's calcium warehouse, and realized that calcium wasn't just structural, it was a messenger. Historians still argue whether the early muscle studies or the later imaging techniques truly cracked the secret. Either way, calcium went from a dull mineral to a star player. In your heart, calcium handshakes keep the rhythm steady. Each beat starts with a wave of ions flooding into cardiac muscle cells, triggering a synchronized contraction. Without it, the beat stutters or stops. It's eerie to think your heart's relentless drum depends on the same element that also strengthens your bones. Calcium is multitasking at a cosmic level: scaffold, signal, and sometimes executioner. Yes, executioner, because too much calcium in the wrong place can trigger apoptosis, the polite form of cell suicide. The ions pour in, mitochondria open their gates, and the cell quietly dismantles itself. It's strangely poetic. The same signal that lets your muscles dance can also whisper the command to die. Here's a quirky tidbit. Jellyfish use calcium-triggered proteins to glow. The green fluorescent protein, first isolated from Equoria victoria, lights up when calcium floods in. That discovery didn't just explain bioluminescence. It gave scientists a glowing marker to track cellular activity. Entire Nobel Prizes have been handed out because someone thought, hey, why is that jellyfish lighting up? Now, labs around the world watch calcium flicker in real time, like seeing your cells. Private fireworks show. You drift closer to a neuron and watch the calcium cascade at a synapse. An electrical signal arrives, calcium channels open and the ions flood in. Within milliseconds, neurotransmitter vesicles fuse with the membrane, spilling their contents into the synaptic cleft. That's how thoughts jump from one cell to the next. Without calcium, your brain would go silent. No memory, no motion, no whispered as you fall asleep. Scholars still debate the full extent of calcium's signaling versatility. Some argue it's the universal second messenger, the punctuation in every cellular sentence. Others say its role is exaggerated, that calcium is just one of many messengers and not uniquely special. But the sheer drama of its effects, heartbeat, thought, death, makes it hard not to treat calcium as the diva of intracellular communication. You notice the elegance of its control. Calcium isn't left to drift freely. It's stored in organelles, buffered by proteins, released in pulses. Too much would be chaos. Too little would be silence. The cell walks a tightrope, keeping the balance like a drummer tapping just hard enough to keep the band in sync without drowning them out. And of course, calcium doesn't act alone. It partners with calmodulin, a protein that hugs calcium tightly and activates dozens of enzymes. Together, they're like a dynamic duo, Batman and Robin in molecular form. The humor, of course, is that calmodulin looks nothing like a superhero. It's more like a floppy ribbon that stiffens when calcium hugs it. But looks don't matter. The signals roar on. You chuckle at another thought. Every time you get startled and your muscles tense, every time your eyelids flutter as you drift into REM sleep, that's calcium gossiping in the dark. It's a language so fast, so fleeting, you'd miss it if not for the glow of special dyes in a microscope. Yet it's happening right now, as you lie there listening. You drift away from the flickers, the sparks dimming like a campfire going quiet. Calcium has passed its message along, and the cell resumes its hum. The handshakes are over for now, but the memory of their brightness lingers. Sharp, silent punctuation marks in the endless conversation of your cells. The flickers of calcium fade into the background, and now you feel yourself carried outward, into the larger currents that flow across the whole body. Here, the messages aren't confined to one cell whispering to another. They're telegrams, hormones traveling long distances, drifting like chemical postcards in the bloodstream. They're slower than electrical impulses, less flashy than calcium sparks, but their words carry weight. A few molecules of the right hormone can flip your mood, change your metabolism, or decide whether you feel hungry enough to raid the fridge at midnight. You watch the bloodstream like a postal service. Molecules of adrenaline, epinephrine if you're on the formal side, shoot through the vessels after being secreted from the adrenal glands. They bind to receptors on heart cells, urging them to pump harder, faster. A few drops of hormone, and suddenly the whole body feels the rush. That's chemical poetry, tiny signals magnified into pounding heartbeat and trembling fingertips. Historically, the concept of hormones as messengers dates back to the late 19th and early 20th centuries. One famous story. In 1902, scientists Bayliss and Starling showed that a mysterious substance, later named secretin, could trigger digestive juice production even when nerves were cut. It was the first solid evidence that chemicals could deliver messages through the blood. Historians still argue whether their experiment marked the true birth of endocrinology, but either way, hormones earned their title: internal secretions carrying coded instructions. You notice insulin molecules now tiny couriers from the pancreas. They dock onto receptors on muscle and fat cells, telling them to pull glucose from the blood. Without this system, sugar lingers too long, leading to the chaos of diabetes. Insulin is one of the most famous hormonal telegrams, a gentle tap on the shoulder. Hey, cell, time to stock the pantry. Then comes cortisol, drifting like a slow-burning fuse. Released during stress, it adjusts metabolism, suppresses immune responses, and generally keeps you on high alert. Useful in moderation, exhausting when constant. You can almost imagine your cells groaning. Another cortisol memo. Does the boss know we haven't even finished yesterday's adrenaline order? Here's a quirky tidbit. Oxytocin, often called the love hormone, surges during hugs, childbirth, and even when petting a dog. But it isn't all hearts and rainbows. In some contexts, oxytocin can actually increase envy or tribalism. The same chemical that bonds you to your baby can also make you suspicious of outsiders. Chemistry is nuanced like that, romantic one moment, petty the next. Hormones don't just send orders, they time them. Melatonin trickles out from the pineal gland as night falls, gently nudging you towards sleep. Growth hormone surges in pulses, especially during those deep slumbers when children sprout taller. Thyroid hormones set the overall pace, like a conductor tapping the tempo for the entire orchestra. Too little and everything drags sluggishly. Too much, and it's a frantic, jittery crescendo. Scholars still debate how best to define hormones. Are they only molecules secreted into blood by glands? Or does the definition include local messengers, paracrine signals whispered between neighboring cells? The edges blur and the dictionary wavers, but the principle remains. Hormones are your body's telegrams, chemical sentences that turn isolated cells into a coordinated you. You can't help but chuckle at the thought of hormones as workplace memos. Adrenaline, emergency drill, everyone to high alert. Insulin, lunch delivery is here, grab glucose while it's hot. Melatonin, lights out folks, tomorrow's another shift. And cortisol, the overzealous manager who keeps sending emails marked urgent even at 3 in the morning. The bloodstream around you feels like a river of overlapping letters, love notes, warnings, pep talks, and reminders. Each one is addressed to specific receptors, ignored by others, delivered precisely where needed. You realize that while your conscious mind crafts words and sentences, your body has been exchanging chemical prose all along, elegant and invisible. As you drift onward, you notice how slow the hormones feel compared to ions and neurotransmitters. They're less like instant messages, more like handwritten letters mailed across town. But sometimes that slowness is the point. A sprint needs lightning-fast calcium and nerve chatter. A pregnancy requires months of hormonal choreography. Both are communication. The difference is packing. You leave the river of hormones behind, but the feeling lingers. That your body is a city not just of whispers and sparks, but also of telegrams carried across long distances, making sure every neighborhood knows what's happening. Whether it's hunger, stress, or the quiet hush of sleep, the language of hormones is the thread weaving your whole self into one story. The hormonal telegrams fade into the distance, and now you're pulled toward a new kind of chatter, sharper, more suspicious, like a neighborhood watch group that never sleeps. You've arrived in the immune system's territory, where cells don't just whisper. They challenge, demand passwords, and sometimes throw punches. This is immune code, the biochemical language that decides who belongs and who doesn't. You hover beside a T cell, sleek and purposeful. On its surface, receptors twitch, scanning passing proteins like bouncers checking IDs. Every healthy cell in your body carries a molecular badge, a set of markers called MHC molecules. I'm one of us, they declare, showing a sample of what's going on inside. A T cell brushes past, reads the badge, and nods. All clear. But if the badge looks wrong, say, hijacked by a virus, the T cell raises the alarm. Historically, the discovery of this recognition system reshaped biology. In the 1950s and 60s, researchers realized that tissue rejection after transplants wasn't random. It was immune cells spotting non-self markers. Historians still argue whether the credit belongs more to immunologists who studied grafts or geneticists who mapped the MHC region. But either way, it cracked the code of cellular identity. You drift into a lymph node, a bustling checkpoint where immune cells crowd together like gossipers at a town square. Antigen-presenting cells wave fragments of pathogens like wanted posters, and the T cells gather to study them. Does this look familiar? One cell seems to ask. Yes, I've seen this flu strain before, replies another, and the order goes out. Mobilize. B cells get the message too, firing up their factories to churn out antibodies, Y-shaped proteins that zip into circulation, tagging invaders for destruction. Here's a quirky tidbit. Some scientists describe antibodies as sticky notes for the immune system. They don't always kill invaders themselves. They just label them, like putting a fluorescent sticker on a suspicious package. The humor writes itself. You imagine a diligent B cell running around, plastering neon, kick ME notes onto every bacterium in sight. The code words extend further. When cells are infected, they release cytokines, chemical alerts that rally reinforcements. Some cytokines call in fever, raising your body temperature to make life harder for microbes. Others summon inflammation, swelling tissues and turning them red. It's messy, but effective. Too much, though, and it spirals. Think of a cytokine storm, where the immune system floods the body with signals so intense that the response becomes dangerous in itself. Scholars still debate whether cytokine storms are overreactions gone rogue, or just extreme versions of normal defense. You notice how precise the communication is. Natural killer cells patrol like silent ninjas, checking for the right molecular handshakes. If a cell fails to flash its badge, the killer strikes, releasing perforins that punch holes in the membrane. It's brutal but efficient, and all of it depends on biochemical code. Badges, signals, and passwords too complex for the naked eye but perfectly fluent to your immune cells. Then there's memory, a special kind of immune gossip that lingers. Once your body fights off an infection, memory B and T cells stick around, remembering the code words. The next time the same pathogen shows up, the response is faster, sharper, almost smug. That's why vaccines work. They teach your cells the code in advance, so when the real intruder arrives, your defenses already know the password. But there's a shadow side. Sometimes the system misreads the code. Allergies, for example, are overreactions to harmless signals, pollen mistaken for an intruder, peanuts treated like pathogens. The immune system throws the full alarm and you're left sneezing, swelling, or worse. Autoimmune diseases are another betrayal when cells mistake self for non-self. Rheumatoid arthritis, lupus, type 1 diabetes, cases where the password is correct, but the guards don't believe it. Even here, scholars still argue whether autoimmunity is a programming glitch, a miscommunication, or some ancient survival strategy gone awry. You chuckle at the thought. If your immune cells were running an online forum, they'd be the strictest moderators imaginable. Post the wrong phrase and you're banned immediately. Accidentally use the wrong emoji and the entire thread gets deleted. Overzealous, perhaps, but that vigilance is why you're still alive to laugh about it. The lymph node buzzes with activity and you drift back, letting the code words swirl around you. Every handshake, every antigen presentation, every sticky antibody note. It's all part of a language so old and so precise that entire medical fields revolve around deciphering it. The immune system isn't perfect, but it's relentless, fluent in its own shorthand. Without it, the microbial world outside would have already rewritten your story. You leave the bustling immune forums behind, only to sense a quieter drama unfolding nearby. The chatter dims, the signals soften, and you realize you're drifting into the solemn space of a cell preparing for its final act. Not the violent rupture of injury or infection, but a graceful bow out. This is apoptosis, programmed cell death, a farewell so polite it feels choreographed. The cell's outer membrane begins to bubble slightly, as if exhaling sighs. Inside, the nucleus condenses, DNA neatly chopped into manageable pieces. No screaming, no chaos. Just an orderly dismantling with enzymes carefully disassembling the machinery. The fragments will be packaged into little vesicles and gently recycled by neighboring cells. If cellular life is a constant conversation, apoptosis is a quiet goodbye note, folded with care. Historically, apoptosis wasn't formally named until the 1970s, though scientists had noticed dying cells long before. The term comes from Greek, meaning falling off, like leaves drifting from a tree. Historians still argue whether earlier anatomists fully grasped its elegance or just saw it as ordinary decay. What's certain is that apoptosis changed how we understand life. Death isn't always failure. Sometimes it's a message, a necessary punctuation mark in the ongoing sentence of growth. You drift closer and watch proteins called caspases snap into action. They're executioners by trade, cleaving other proteins, dismantling the cell from within. Yet there's nothing gruesome about it. It feels almost ceremonial. Imagine librarians carefully shredding outdated books, making room for new editions. The caspases aren't cruel. They're efficient, ensuring that no mess lingers behind. There's humor here too if you squint. Some biologists jokingly call apoptosis cellular suicide with manners. Unlike necrosis, which is messy and inflammatory, apoptosis is tidy. The neighbors barely notice. No alarm bells, no swelling. It's like a roommate moving out quietly in the night, leaving their keys on the counter and a thank you note in the fridge. A quirky tidbit surfaces. In developing embryos, apoptosis sculpts the body's shape. Your fingers and toes, for instance, began as paddles. It was apoptosis that cleared the webbing, carving digits from a block of tissue. Without it, you'd still have mitten hands. Scholars still debate whether certain evolutionary oddities, like webbed feet in some animals, reflect subtle tweaks in apoptotic timing. The idea that death shapes form is oddly poetic. The cell you're watching begins to fragment, little blebs pinching off like soap bubbles. Neighboring macrophages approach, ready to engulf the pieces. There's no mourning here, no tragedy, just recycling. The building blocks will be reused, woven into new proteins, new membranes, new chatter. In death, the cell's voice doesn't vanish. It just changes register, contributing to the chorus in another form. Apoptosis has a darker side too. Cancer cells often evade it, refusing to heed the signals that should cue their exit. It's like someone ignoring the polite closing time music in a bar and insisting on staying forever. On the other hand, excessive apoptosis can thin tissues, contributing to degenerative diseases. Balance is everything. Too little and cells linger dangerously. Too much and systems weaken. You find yourself thinking, if cells had personalities, some would leave with dramatic speeches, some would cling to the doorframe, and others, like this one, would quietly vanish mid-sentence, graceful and unassuming. The quiet cell death is almost more moving than the bustling chatter of life. And here, where scholars still debate, is apoptosis purely mechanical, or does it reflect something deeper about how life values order? The process feels intentional, though it's just chemistry. Yet the symbolism lingers. The idea that death can be beautiful, structured, and beneficial is hard not to interpret as a kind of wisdom written into molecules. The last fragments of the cell dissolve into its neighbors, leaving no trace of drama. The chatter resumes uninterrupted as though nothing happened, but you know better. That goodbye wasn't silence, it was part of the language. Cells speak not only in signals of growth and survival, but also in departures. And somehow, that makes the symphony of your body feel even more whole. The quiet farewell of apoptosis fades behind you, and now you're pulled backward in time, to an era when your body wasn't yet you at all. You find yourself in the embryo, where cells are multiplying like wildfire and holding the most intense conversations of their lives. The chatter here is urgent, packed with decisions. Who will become skin? Who will turn into bone? Who will fashion the swirl of your inner ear? It's embryonic Morse code, bursts of signals, dots and dashes of chemical language, spelling out the blueprint of a future human. The first thing you notice is the gradients. Proteins and growth factors spread across the embryo in waves, forming invisible maps. Depending on how much of a signal a cell receives, it adopts a different fate. A little of this molecule, you're a spinal cord. A lot of it, congratulations, you're now part of the brain. The precision is staggering. One cell's interpretation of a chemical ... might become the difference between an eyeball and an elbow. Historically, this field blossomed in the early 20th century, when embryologists like Hans Spemann experimented with tiny amphibians. By transplanting bits of tissue, he discovered an organizer region that could direct the fate of surrounding cells. Historians still argue whether his Nobel Prize in 1935 should have been shared more widely, but the principle remains clear. Embryos are guided not by chaos, but by conversations written in chemical code. You drift closer and watch Notch signaling at work. One cell juts out a molecular hand, the Notch receptor meets its ligand neighbor, and the dialogue begins. It's like two kids drawing a chalk line between their yards. You be nerve, I'll be skin. This back and forth ensures that cells don't all choose the same fate at once. Without it, you'd end up a shapeless blob of identical tissue, more pancake than person. Here's a quirky tidbit. Fruit fly embryos have stripes of gene expression that look like tiny barcodes under a microscope. These stripes, created by signals like the famous hedgehog genes, lay out the segmented body plan. Yes, biologists really did name a gene Sonic Hedgehog after the video game character. Imagine your spinal cord being patterned thanks to a molecular shout-out to a blue cartoon hedgehog. Science has a sense of humor, even when it's busy building vertebrates. The embryo around you is alive with chatter. WNT signals wash over clusters of cells, bone morphogenetic proteins whisper structural instructions, and FGF molecules sprint across tissues like impatient messengers. The result is choreography on a grand scale. Limbs bud, organs sprout, the neural tube folds. Each move is encoded in patterns of dots and dashes, bursts of presence and absence. Yes, and no. And yet, scholars still debate the balance of fate versus flexibility. Are embryonic cells rigidly following a script written in gradients, or do they improvise based on the chatter of their neighbors? Some argue that development is deterministic. Others insist it's adaptive, with wiggle room for variation. Either way, the outcome is miraculous. From a single fertilized egg, a galaxy of specialized cells emerges, each fluent in its dialect of the hidden language. You pause at the forming hand. At first it looks like a paddle, a smooth blob, but then waves of apoptosis sweep through, carving spaces between fingers. It's the same quiet goodbye you saw earlier, now transformed into an artistic tool. Death here is part of creation. The Morse code isn't just building, it's sculpting. Imagine if you could hear it translated. Cell A, you're a neuron, connect to that one over there. Cell B, your muscle, line up in parallel. Cell C, sorry, we don't need you, make way for fingers. The chatter is relentless, and yet the embryo doesn't stumble. Each signal is received, decoded, and acted upon, building complexity without ever stepping back to admire the whole. You chuckle at the thought. If embryos had group chats, they'd be the most chaotic ones ever created. Thousands of participants, half the instructions contradictory, yet somehow the plan still gets executed flawlessly. And the moderators, just gradients of molecules diffusing through jelly-like tissue, keeping order through nothing more than proximity and timing. The embryo grows larger as you drift, the Morse code crackling around you like invisible telegraphs. The signals don't shout, they pulse, ebb, and ripple, sculpting tissues with subtlety. It's a reminder that even your very first days were built on conversations, tiny, chemical, unending. The chatter was your origin story, and though you'll never remember it, your body carries its echoes in every cell. The embryonic Morse code dissolves around you, and suddenly the chatter sharpens into crackling sparks. You're gliding into the realm of neurons, where communication takes on a different rhythm, fast, glittering like fireflies blinking across a summer field. These are synapses, the tiny gaps where nerve cells talk to each other, and the language here is part electricity, part chemistry, all urgency. A neuron fires and you feel it ripple. An electrical wave racing down the axon, sodium and potassium gates clicking open and shut like dominoes tipping in sequence. When the wave hits the synaptic terminal, calcium floods in and vesicles snap into action. They fuse with the membrane and spill their contents, neurotransmitters, into the synaptic cleft. It's a blink, a spark, a whispered word carried on molecules. On the other side, receptors wait. Dopamine, serotonin, glutamate, GABA, each neurotransmitter fits into its receptor like a note played on a specific instrument. Some excite the next neuron, urging it to fire too. Others calm it down, whispering, "Not yet." The balance of these voices shapes everything from your heartbeat to your mood. Even now, as you listen in this half-dreaming state, your neurons are trading sparks to sustain the thought of drifting off. Historically, synapses were once thought to be direct electrical connections. It wasn't until the late 19th and early 20th centuries that scientists realized chemicals were involved. Charles Sherrington coined the word synapse in 1897, describing the junctions as places of integration. Historians still argue whether Sherrington truly foresaw the complexity of chemical neurotransmission, but his metaphor stuck. The synapse became not just a gap, but a stage for communication. You drift closer to one and the scale is breathtaking. The cleft is only about 20 nanometers wide, so small you'd need 5,000 of them to span the thickness of a sheet of paper. And yet, across that tiny gulf, the entire theater of thought plays out. Your first kiss, your favorite song, your stressed dreams about forgetting a password, all of it happened because molecules hopped this gap. Here's a quirky tidbit. In the 1920s, Otto Lowy proved neurotransmitters existed with a dream-inspired experiment. He stimulated a frog's vagus nerve, slowed its heart, then transferred the fluid to another frog's heart, which slowed too. The mysterious substance was acetylcholine. Lowy reportedly woke up one night with the idea, scribbled it down, and nearly lost the note by morning. Imagine if he'd rolled over and said, I'll remember later." Entire neuroscience textbooks might be empty. The conversation at the synapse isn't tidy. Neurotransmitters spill, get reabsorbed, broken down, recycled. Drugs exploit this. SSRIs, for example, block the reuptake of serotonin, letting more linger in the cleft, nudging mood upward. Caffeine blocks adenosine receptors, tricking neurons into staying alert. Cocaine floods dopamine synapses, overwhelming the system. It's all chemical eavesdropping, hacking the language of sparks and whispers. You chuckle at the thought. If neurons had social media, dopamine would be the like button. Every time a post gets attention, dopamine signals reinforce the behavior. It's the original feedback loop, older than civilization, running on molecules hopping across microscopic gaps. Scholars still debate whether consciousness itself emerges from this synaptic chatter. Is your mind just the sum of billions of these sparks, or is there something more? The argument rages on, mechanical explanation versus mystery. But meanwhile, your synapses keep firing, indifferent to philosophy. You watch a burst of glutamate cross a cleft, exciting the next neuron into action. A moment later, GABA counters with inhibition, like a friend gently pulling someone back from dancing on a table. The push and pull is constant, keeping your brain from spiraling into chaos. Balance is everything. Too much excitation and you risk seizures. Too much inhibition and the lights dim into stupor. The beauty of it all hits you, the blinking sparks, the whispered molecules, the ceaseless exchange. Every thought you've ever had was born here, in synaptic secrets too small to see without powerful microscopes. And now, as you drift in this story, the same language is crafting the dreamlike images in your mind. Your neurons are talking, sparking, whispering, writing you into existence one molecule at a time. The firefly sparks of the synapses fade behind you, and you drift into a quieter, more ominous space. Here the language isn't elegant or efficient. It's garbled, scrambled, like a group chat filled with typos and autocorrect disasters. This is miscommunication at the cellular level, and it's the birthplace of some of the body's greatest tragedies. You hover over a cluster of cells dividing rapidly. At first it looks normal, mitosis ticking along, chromosomes neatly lined up, membranes splitting. But then you notice the signals have gone fuzzy. Growth factors are firing non-stop, even though the cells aren't supposed to multiply here. The brakes, the apoptotic cues you saw earlier, are ignored. This is cancer, the cellular equivalent of someone replying "party at my place" to every text, until the entire neighborhood floods into one overcrowded house. Historically, the connection between miscommunication and cancer became clear in the 20th century, as scientists discovered oncogenes, genes that, when overactive, shout "divide" too loudly. Tumor suppressor genes, on the other hand, normally whisper "slow down," but when silenced, their cautionary voice is lost. Historians still argue whether the discovery of oncogenes or tumor suppressors was the bigger breakthrough, but both made one fact clear. Cancer is less about rogue cells and more about corrupted conversations. You drift onward and see another type of miscommunication, an allergy. Here, harmless pollen proteins bump into immune cells, but instead of a calm nod, the system panics. Mast cells burst, releasing histamine, swelling tissues, clogging sinuses. It's a fire alarm blaring just because someone microwaved popcorn. Funny in theory, miserable in practice. Then, deeper still, you encounter autoimmunity. Cells glance at healthy tissues and somehow mistake them for intruders. Antibodies tag joints in rheumatoid arthritis, insulin-producing cells in type 1 diabetes, nerve linings in multiple sclerosis. It's as if your immune moderators suddenly turned paranoid, banning longtime members for imaginary offenses. Scholars still debate the root cause: genetics, infections, sheer bad luck. Whatever the trigger, the result is tragic: your own cells becoming enemy code. Quirky tidbit: there's a theory that friendly fire from the immune system might occasionally bring evolutionary benefits. For instance, certain autoimmune-related genes appear to protect against infections like tuberculosis. It's the strange irony of biology. Even destructive miscommunications sometimes leave useful ripples in the gene pool. Miscommunication isn't limited to disease. Even everyday feelings can trace back to scrambled signals. Depression, for example, often links to imbalances in neurotransmitter chatter. Too little serotonin here, too much glutamate there. Fatigue, brain fog, and stress flare when hormones mistime their releases. Your cells aren't plotting against you. They're just trying to talk with faulty microphones and static-filled lines. You chuckle at the thought. If your body had a customer service desk, miscommunication would be the #1 complaint. Excuse me, why are my mast cells acting like pollen is a terrorist? Hello, yes, my T cells keep attacking my pancreas, can I get a refund? The absurdity doesn't erase the seriousness, but humor has always been the brain's way of filing the paperwork for chaos. The strangest part is how small the errors can be. A single misread codon in RNA, one protein folded slightly wrong, one receptor misfiring, and the ripple spirals outward, magnifying into systemic collapse. It's the equivalent of adding one wrong letter to a sentence and ending up with an entirely different story. And still, scholars debate whether miscommunication is the inevitable price of such a complex language. With billions of cells and countless signals flying every second, maybe glitches are unavoidable. The marvel isn't that things go wrong, it's that they go right as often as they do. Most of the time, your cells are fluent. Miscommunication is the rare typo in a manuscript that otherwise holds together. You drift past the rogue cancer cells, past the sneezing immune responses, past the paranoid antibodies, and back into calmer waters. The garbled signals linger in your mind, though. They're a reminder that language, whether human or cellular, carries risk. Meaning can be lost, twisted, or ignored. And when it happens inside you, the consequences aren't just confusion. They're the fine line between health and illness. The garbled chatter of miscommunication drifts away, and now a new chorus rises. Unexpected, rowdy, and not entirely yours. You're moving into the gut, where trillions of microbial voices hum together. This isn't just digestion, it's dialogue. Your microbiome, an entire ecosystem of bacteria, fungi, and archaea, sends messages that ripple through your body like small talk at a crowded cafe. At first glance, it looks chaotic. Microbes cling to the intestinal lining, forming dense neighborhoods. They trade metabolites, release chemical signals, and even produce neurotransmitters. Serotonin, for example, yes, the feel-good molecule you associate with mood, is largely produced in the gut, not the brain. The idea that your happiness partly depends on bacterial gossip is both unnerving and hilarious. Historically, this microbial chatter was ignored. For decades, gut bacteria were seen as background noise, useful for digestion, maybe, but hardly central. Then in the late 20th and early 21st centuries, studies began linking the microbiome to obesity, immunity, and even mental health. Historians still argue whether the hype has outpaced the evidence, but one thing is certain: your microbes aren't silent. They're fluent in biochemistry. You watch as one bacterium produces a short-chain fatty acid, butyrate. Nearby colon cells absorb it gratefully, using it as fuel. Another bacterium synthesizes vitamin K, a molecule your body doesn't make on its own. Yet another churns out molecules that tweak immune responses, teaching T cells to stay calm instead of attacking every passing crumb. It's community theater, each microbe playing its part. And then comes the quirky stuff. Some microbes release chemicals that interact with the vagus nerve, the main communication line between gut and brain. In mice, altering gut bacteria has been shown to change anxiety levels and even social behavior. Imagine your bacteria sliding into your DMs. Hey, we made you crave chocolate today. Also, maybe call your mom. Scholars still debate just how direct these effects are in humans. But the conversation is loud enough that the idea of psychobiotics is gaining traction, probiotics marketed to improve mood. You drift through this microbial metropolis, and the small talk takes on a rhythm. Signals bounce not only between microbes and cells, but also microbe to microbe. Quorum sensing, bacteria counting their own numbers by releasing signaling molecules, lets them decide when to act. At low levels, they stay quiet. At high levels, they switch on genes for group behaviors like forming biofilms. It's basically a bacterial group chat. We've got quorum time to build a fortress. Of course, the chatter isn't always friendly. Pathogens slip in, trying to shout over the crowd. Some microbes manipulate host signals to their advantage, hacking your chemical language to carve out space. Others fight each other, releasing toxins that silence competitors. Your gut isn't just a dinner party, it's a turf war, and you're the landlord who barely notices. Here's a quirky tidbit: fecal transplants, yes, exactly what it sounds like, can restore microbial chatter in people whose microbiomes have been devastated by infection. It's both absurd and astonishing. Borrow someone else's bacterial language, and your own health can be rebooted. Who knew swapping gossip could be so literal? You chuckle at the thought. If microbes had personalities, some would be health nuts, constantly pushing kale smoothies and exercise reminders, while others would be chaos gremlins whispering, "Ice cream for dinner, trust me." And the worst part, sometimes the gremlins win. Cravings, moods, even your social energy may be influenced by the chatter of cells you don't technically own. Scholars still argue how much control the microbiome really has. Is it the puppeteer pulling strings on your brain and body, or just a noisy roommate whose voice occasionally slips into your inner monologue? The science is young, but the fact remains. Their chatter is part of your language now. You are not alone in your own body. The microbial hum fades as you drift upward, leaving behind the busy chatter of butyrate, serotonin, and quorum calls. But the realization lingers. Your cells don't just talk to each other. They host an entire rented choir, microbial tenants whose small talk shapes the melody of your life. The microbial chatter dissolves behind you, and now the tone shifts. The voices of your cells feel older, wearier, like records playing with a faint crackle. You've entered the realm of aging, not a sudden breakdown, but a slow misinterpretation of the language that once sang so clearly. Over time, the grammar falters, the punctuation blurs, and your cells start winging it. You notice a fibroblast cell nearby, once spry and chatty, now sluggish. Its signals to make collagen are faint, and the surrounding extracellular matrix begins to sag. This is why skin wrinkles, not because the cell gave up, but because its voice has grown hoarse. Collagen once snapped taut like fresh elastic. Now the strands are patchy, frayed, and the conversation about firmness is a little mumbled. Historically, the science of aging has swung between theories. In the 1950s, Denham Harman proposed the free radical theory, that oxidative damage from reactive molecules gradually garbles cellular language. Later came the telomere theory. Protective DNA caps short each time cells divide, like frayed shoelaces, until they can't keep messages intact. Historians still argue whether any single theory explains aging, or if it's simply the sum of every communication glitch piling up. You drift closer to a dividing cell and notice its telomeres, tiny protective ends of chromosomes. They've shortened with each replication, and now the cell hesitates. Divide again, or slip into senescence, a limbo where it still lives but no longer contributes. Senescent cells don't just go quiet. They sometimes mutter strange, inflammatory signals that confuse their neighbors. It's like an old radio stuck between stations, crackling noise that disrupts the symphony. Quirky tidbit: some scientists nicknamed senescent cells zombie cells. They don't die, but they don't function properly either. Instead, they linger, spreading confusion and sometimes coaxing nearby cells into dysfunction. Imagine trying to focus on your work while someone in the office corner loudly plays accordion polka all day. It's distracting, and eventually the whole office slows down. You notice mitochondria too, the powerhouses you visited earlier. Their membranes are less tidy now, their DNA accumulating damage. The electron transport chain, once a smooth assembly line, sputters, leaking reactive oxygen species. The gossip that once fueled life now sometimes sparks little chemical fires, not enough to kill the cell outright, but enough to garble messages. And yet the picture isn't only decline. Some cells retain resilience, compensating for the faltering signals of their neighbors. Stem cells, for example, lie dormant in tissues, ready to wake and replace damaged cells. But even stem cells tire with age, their responses slower, their chatter hesitant. Scholars still debate whether aging is inevitable entropy or a program the body follows intentionally, an ancient clock ticking down. Hormonal telegrams weaken too. Growth hormone pulses less frequently. Sex hormones dwindle. Melatonin's nightly whisper grows faint, fraying sleep cycles. The body still receives the messages, but it's like straining to hear a loved one calling from across a windy field. Misunderstandings pile up. And yet here comes the humorous side. Some aging cells don't just miscommunicate, they overcommunicate. Inflammation, sometimes called inflammaging, rises with age, as immune cells send too many alerts. It's like the group chat where older relatives reply K 20 times in a row, flooding the thread with noise. Not harmful individually, but collectively exhausting. A quirky human-level twist. Calorie restriction in animals often slows aging, possibly by reducing metabolic chatter and oxidative noise. It's as if the less you feed the party, the more polite the conversations become. The debate rages on whether the same applies to humans, but the idea that silence extends life has a strange poetry. You float back and see the larger picture. Aging isn't a single failure, it's a conversation gradually losing clarity. The elegant ballet of signals you saw earlier becomes a little clumsier, the timing a little off. A protein misses its cue, a receptor responds too slowly, a hormone memo arrives too late. Individually, these slips are minor. Together, they build into stiffness, fatigue, and fragility. And yet there's grace in it. The language still flows, even if a little garbled. Your cells don't stop speaking. They improvise, filling gaps, leaning on each other, whispering in half sentences. Aging is less about silence than about reinterpretation. The chatter continues, softer now, but no less alive. The weary voices of aging cells drift behind you, and now the soundscape changes again. The chatter is still molecular, still microscopic, but you sense new listeners, humans leaning in with tools, wires, and glass lenses. Scientists have learned not only to overhear the cellular language but to speak back. This is artificial eavesdropping, and it's both wondrous and a little unnerving. You float above a cluster of researchers peering through powerful microscopes. Fluorescent tags glow like neon signs, marking where proteins fold, where calcium flickers, where DNA unspools. Each flash is a syllable of the cellular dialect, now visible thanks to technology. Green fluorescent protein, the same jellyfish trick you saw earlier, has become a translation device, letting humans watch cells gossip in living color. Historically, the art of eavesdropping began with simple stains and dyes in the 19th century. Scientists dunked tissues in colorful chemicals to reveal structures under a microscope. But the real revolution came in the late 20th and early 21st centuries, with gene sequencing and CRISPR. Historians still argue whether the double helix discovery in 1953 or the CRISPR breakthrough in 2012 was the greater leap, but both cracked open the possibility of not just observing the language of cells, but editing it. CRISPR especially feels like a cheat code. It lets scientists target specific DNA sequences and snip them with pinpoint accuracy. Want to silence a gene? Insert a new one. It's like whispering directly into the nucleus, rewriting the librarian's catalog without waiting for permission. Quirky fact: CRISPR wasn't invented by humans at all. It's borrowed from bacteria, who used it as an immune memory system against viruses. Humans just figured out how to hijack it for our own purposes. Classic scientific eavesdropping. You drift closer to a lab bench and see scientists growing organoids, miniature, simplified versions of organs cultivated in dishes. They watch how cells organize themselves, signaling in little embryonic whispers, forming tiny brains, hearts, or kidneys. The surreal part: these mini organs chatter too, and researchers are learning to decode their dialects. It's like raising little cellular neighborhoods in petri dishes, just to overhear what they're saying. Of course, meddling with the language comes with risks. Edit one gene and unintended consequences ripple out. You might silence a disease pathway but accidentally confuse another system. It's like changing one line of dialogue in a play and finding the whole plot spirals into absurdity. Scholars still debate the ethics of how far to push this. Should we cure genetic disease, enhance traits, design Bobbies fluent in an optimized version of the cellular language? The questions hover, unanswered. Here's a lighter tidbit. Some researchers have literally encoded secret messages into DNA. In one experiment, they spelled out words, even music, inside the genome of bacteria. The cells carried these Easter eggs like living USB drives, whispering not just their own language but human jokes embedded in molecular code. It's the most literal kind of eavesdropping. You're not just listening to the conversation, you're adding your own punchlines. You drift through another lab, where scientists are developing drugs to mimic or block signals. Immunotherapy, for instance, trains immune cells to recognize cancer's disguises. It's like giving the body a dictionary of enemy code words. Meanwhile, neuroscientists experiment with optogenetics, using light to control neurons by inserting light-sensitive proteins into their membranes. Suddenly, you can flick neurons on or off with a laser pointer. Imagine hijacking a synapse mid-conversation, forcing it to laugh or fall silent. And then there's synthetic biology, the boldest eavesdropping of all. Scientists design new genetic circuits, reprogramming cells to act like tiny computers. Some engineered bacteria glow when they detect toxins, effectively becoming living sensors. Others are tweaked to produce drugs inside the body, whispering therapeutic messages where they're needed most. You can't help but marvel at the audacity. Humanity isn't just listening to the language of cells, we're starting to co-write the script. Scholars still debate where the line should be drawn. Are we translators, helping cells say what they always meant to say more clearly? Or are we loud interlopers, drowning out their native tongue with our edits and hacks? The conversation in scientific journals is as heated as any cellular gossip. But the fact remains. The eavesdropping is happening, and the whispers of your cells are no longer private. You drift back, leaving the glow of microscopes and petri dishes behind. The memory of it lingers, though. The idea that somewhere right now, someone is watching your cells whisper and maybe even whispering back. The language isn't hidden anymore. It's being recorded, translated, rewritten, and the story of life itself is becoming a collaboration. The glow of lab lights fades, and you find yourself back inside the body, floating among the chatter you've come to know so well. Only now, a question hangs in the air, heavier than all the whispers combined. Are these signals just chemistry, or is there something more? You've been listening to molecules spark and fold, to proteins gossip and mitochondria hum, but tonight the question lingers like an echo. Are cells aware of the language they speak? You watch a neuron firing, calcium surging in, neurotransmitters spilling out, receptors snapping shut on the other side. It looks mechanical, precise, almost too perfect. But zoom out far enough, and this cascade is what creates your thoughts, your memories, your sense of you. Scholars still debate whether consciousness is simply the sum of these conversations, or whether something ineffable emerges from them. Are you just a byproduct of chatter, or is the chatter itself aware? Historically, biologists have been cautious here. In the early 20th century, talk of cellular intelligence was dismissed as mystical nonsense. Cells were machines, nothing more. But even then, some fringe scientists wondered if life at the smallest level had a spark of awareness. Historians still argue whether those musings were pseudoscience or early attempts to grapple with complexity. Today, a few researchers tentatively revisit the idea. Not that cells are conscious in a human sense, but that they might process information in ways that feel eerily intentional. You drift toward the immune system again, remembering how T cells recognize intruders. The specificity, the memory, the way they learn. It feels like decision-making, though it's all molecular. Or consider apoptosis, the quiet cell death you witnessed. A cell receives signals, weighs them against survival cues, and then chooses to die for the greater good. That's not thought in the human sense, but it feels like a choice. Here's a quirky tidbit. Slime molds, single-celled organisms can solve mazes. Place food at 2 points and the mold spreads out, retracts dead ends, and eventually traces the shortest path, like a commuter finding the best subway line. No brain, no nerves, just chemical chatter, and yet it looks like problem solving. Watching it, you can't help but wonder whether awareness is a spectrum, not a binary. You laugh softly at the thought. If your cells were conscious, what would they say about you? Would your neurons gossip about the silly dreams you feed them? Would your gut microbes brag about making you crave pizza? Would your skin cells roll their eyes every time you forget sunscreen? The image is absurd but strangely comforting. The idea that your body is a community, not a monarchy. Scholars still argue, of course. Some insist there's nothing mystical here, just molecules bouncing, following the laws of chemistry. Others suggest that meaning itself, the way cells interpret signals, might be the seed of something like proto-awareness. Not thought, not mind, but a precursor, a quiet hum of being. The debate is unresolved, and maybe it always will be. You float among the cells one last time. The membranes shimmer with whispers, proteins fold like tiny storytellers, mitochondria hum their ancient songs. Calcium flickers, RNA scribbles notes, hormones drift like telegrams. It's a chorus of voices, and whether they know they're speaking or not, the result is undeniable. You, your breath, your heartbeat, your laughter, your sleep. The hidden language of your cells may never tell you outright whether it's conscious or just noise. But as you drift into rest, you realize that doesn't matter. The chatter is steady, the conversation endless, and it carries you gently, molecule by molecule, into the dark, soft rhythm of life itself. And now, as the last whispers of your cells fade into the distance, let your mind quiet too. The microscopic orchestra continues without you needing to notice, each signal flowing seamlessly into the next, keeping you alive in the most unassuming way. You don't have to direct it. You don't have to remind it. The language has been spoken fluently since before you were born, and it will continue speaking as you drift deeper into sleep. Imagine the cells now as faint lanterns glowing in the distance, their chatter softening into a low, steady hum. No more sparks, no more cascades, just the gentle background music of life running smoothly, effortlessly. Every organ, every tissue, every molecule has taken its place in the chorus. And while you rest, the language carries on, tending to every detail without asking for applause. Breathe slowly. Picture your bloodstream as a calm river, no longer rushing but gliding quietly. Picture your neurons dimming their lights, easing into slower rhythms. Picture your mitochondria still humming but softer now, like crickets at dusk. The whole body is settling, translating its chatter into a lullaby meant only for you. Whether cells are conscious or not, they've mastered the art of rhythm, of balance, of continuity. And in their hidden language, the message for you tonight is simple: rest. Allow the chatter to blur into background. Allow your own awareness to fade into dreams. The work is still being done, and you are safe in that steady conversation. So close your eyes now. Let the last of the cellular symphony dissolve into silence and drift gently into sleep.